Welcome to Bio24's lecture on genetically modified organisms, or GMOs. These organisms are products of genetic engineering and are also referred to as transgenic organisms. They're called transgenic, trans meaning move and genic meaning genes. So that means that their genes have been moved around. Usually this is in reference to genes from other organisms being brought into their own DNA. So all of these terms are interchangeable: GMOs, genetically engineered, or transgenic. Today, we're just going to focus primarily on plant genetically modified organisms. And that's because animals that are GMOs are not very common, and with one exception, are not produced for commercial sale. I first want to reference how humans have been genetically modifying organisms for thousands of years. Because when we talk about genetically modified organisms, even though this is specifically in reference to scientists altering the DNA of organisms, this is definitely not the first time humans have altered the DNA of organisms. In fact, for thousands and thousands of years, humans have been artificially manipulating the DNA of many organisms around the planet. This is what's referred to as the process of classic breeding. Classic breeding is a means of altering plant DNA whereby organisms that are comprised of traits considered valuable are retained or kept by the farmer or breeder, and organisms that have traits that are less desirable are destroyed and not allowed to breathe. This is artificial selection. Artificial selection allows humans to manipulate DNA over the course of many generations with a goal in mind, and the goal may always be different. So in the case that you see here, turning the native organism teosinte in South America to what we today refer to as corn, you see that the goal was to produce more kernels, softer kernels, larger kernels, and more tasty kernels. This takes a very long time, often generations, to do something like this. It involves the random mixing of DNA, and with every generation, there is a need to select for the desired traits in the offspring. Also importantly, the only traits that can be bred into or out of the organisms are traits that are already within that species. So for instance, I couldn't make corn taste like fish because corn doesn't have fish DNA in it. Grafting is very different, and that's taking one plant, cutting it at the stem, and attaching it to the rootstock of another plant. Eventually these two will fuse, but the DNA is not shared between them. So here, any fruits that come out of the stem region are going to have just the DNA of the scion or stem portion and will not contain any of the DNA of the rootstock. Likewise, none of the fruits from the stem portion will have any of the traits of the rootstock either. Grafting is really common in the wine industry because grapes are valued for their flavor, and no one wants to interfere with the flavor of a grape just to get a stronger rootstock. And so strong rootstocks are grafted onto really tasty grapes. But again, the DNA in the grapes is not altered in any way. Those grapes would still have very weak rootstocks. Now, all of this has been done for thousands of years, but very recently, in the last few decades, scientists have begun altering DNA in a different way. And that step in altering the DNA of plants is to use genetic engineering, otherwise referred to as plant transformation. Transformation because we're changing the plant shape. and characteristics. Genetic engineering of plants would involve removing, modifying, or inserting genes from one organism into another. By comparison, genetic engineering is much, much faster than classic breeding. It can also involve very precise additions or deletions of DNA sequences, as opposed to classic breeding where the mixing is totally random and unpredictable. And then a huge difference between classic breeding and genetic engineering is that with genetic engineering, you can insert DNA from any organism. You can even artificially make it and insert DNA that's never been found on Earth into a plant cell. The reason why this is possible is because all organisms share a common ancestor. And so all DNA is read exactly the same by every cell on Earth. There is in fact no such thing as a gene, a human gene or an apple gene or a fish gene. It's just that there are genes that were originally found in humans, genes originally found in fish, and so forth. But all cells can read all DNA exactly the same way because we all share a common ancestor. We all have the same language. Now, to generate the DNA that you're going to insert into a plant, you would need to generate what's referred to as recombinant DNA. Recombinant DNA is DNA that's been mixed up by a scientist. There are three essential components to recombinant DNA. The first is the promoter. The promoter can be altered or found from a natural source. And the promoter is what's sort of considered as the on-off switch for the genes in recombinant DNA. They control when and where a gene creates the protein. So here we have a promoter for your gene of interest, and as you can see also a promoter for the marker gene. The most common promoter that's historically used is the promoter found in cauliflower mosaic virus. The reason for this is that this particular virus, though it doesn't infect any people, can infect plants, and when it does, it expresses all of its genes at really high levels no matter what tissue or what time of the year that plant is in. Usually, when a genetic engineer inserts a piece of DNA, it wants that DNA expressed all the time in all tissues. So by using the promoter from cauliflower mosaic virus, you're essentially turning that gene on at really high levels all the time. Secondly, in addition to the promoter gene or the promoter sequence, a genetic engineer will also insert a gene of interest. This is the gene that you want to put into the organism or the plant. Most plants today have genes for either herbicide resistance or pesticides. Now, the herbicide resistance gene of interest is providing the plant with the ability to resist herbicides, specifically Roundup. If you're familiar with Roundup, this is a chemical you can readily buy at a grocery store or garden store, and you can just spray these on weeds in your yard and it'll kill the weeds. It's fairly innocuous as far as herbicides go, harmless, and doesn't harm human health too much, especially compared to other herbicides. And what this gene allows is if a farmer were to insert this gene into a plant, that plant would then not die when sprayed with Roundup. Those plants are referred to as Roundup Ready. There's Roundup Ready corn, Roundup Ready soybean, and others. Another very common gene that's used by genetic engineers and companies and inserted into plants is a gene for pesticides. Specifically, that will kill insects. This gene that's most commonly used is the Bt gene. The Bt gene is a gene that comes from a bacterium, Bacillus thuringiensis. And when consumed by insects, it actually causes the digestive tracts of insects to melt and be destroyed, thus killing the insect. It doesn't work on all insects. Like, for instance, it doesn't do anything to spiders. But in some insects, like weevils, beetles, and some caterpillars, it can cause their death. These Bt plants allow for the growth of the plant to resist insect infestation. There are some plants that are engineered to resist viruses. We'll talk about some of those later. Some plants and animals are engineered to grow faster or larger. Some plants were engineered to have a longer shelf life. And some universities or companies are trying to engineer a sweeter taste or additional nutrients, bigger fruit, or drought or stress tolerance in plants as well. Really, the possibilities are limitless, considering how easy it is to genetically engineer organisms, and that you can also insert totally synthetic genes as well. Once you insert these genes into the organism, how do you know which cells are genetically engineered and which ones are not? This is where that third component of the recombinant DNA comes in, the marker gene. Now, the marker gene is a gene that helps you determine which plants have the gene of interest, which plants have been genetically engineered, and which ones have not. There are two main types of marker genes. The first one of these is an antibiotic resistance gene. Antibiotic resistance genes are genes that are found in bacteria that allow them to resist antibiotics. Now, typically, antibiotics don't affect any other cell type other than bacteria. Except, interestingly, antibiotics, some of them, can end up destroying chloroplasts. And why is that? Well, the reason for this is that chloroplasts, evolutionarily, are actually little tiny bacteria living inside cells. This is also true of mitochondria. This is partly the reason why some antibiotics, when taken by humans, can have their mitochondria affected. Because antibiotics really only destroy the little bacteria living inside cells like mitochondria and chloroplasts, the rest of the plant cell can survive if sugar is provided to it. And that's exactly what you see here in these images. So what happens is after a plant is genetically engineered, the scientist will check to see which plants are genetically engineered and which ones are not by placing the leaves of those plants onto a petri dish with antibiotics. The leaves that can resist the antibiotics are green and stay alive, along with their chloroplasts. And the leaves that cannot resist antibiotics turn bleached white because the chloroplasts die. This means that the white leaves here are not antibiotic resistant and thus do not have the gene of interest and also are not genetically engineered. Whereas the green leaves that you see here are antibiotic resistant, which means that they do have the gene of interest and are genetically engineered. And so then you know that these are your plants that you want to grow in your field. There is another type of marker gene though, and that's the herbicide resistance gene. As we talked about before, this is using Roundup. So you can use Roundup resistance gene as your marker gene as well. The ones that are transformed or genetically engineered would grow fine in Roundup, and the ones that are not would not grow. And so then you'd know which plants are genetically engineered or modified, and you can grow those. In the next section, we'll talk about how scientists get this recombinant DNA into the plants themselves.